Am I audible?
Yeah,
Yes,
you're
ma'am.
audible.
Good evening, everybody. I hope all of you are safe and healthy. At the outset, I wish to place on record my sincere thanks to our publishers, Wolters Kluwer, for organizing this webinar, which is a very important topic, and also for giving me the opportunity to speak in it. So I am Dr. Sumati Murlidhar. I'm professor of microbiology at the Vardhaman Mahavir Medical College in Delhi. And I'm also a consultant microbiologist at the attached Safdarjung Hospital in the Apex Regional STD Center, which is catering to exclusively the diagnosis of STDs and HIV/AIDS. So we also have the ICTC and the SRL, for which I am the in charge, and I'm also the laboratory in charge for the STD and HIV labs. Apart from this, I'm one of the empaneled, lead and technical assessors for assessor for National Accreditation Board for Testing and Calibration Laboratories. That's NABL. And I'm also associated with IGNU for their activities on healthcare waste management. I've written a couple of units for them, and I've also authored and published more than about 55 papers in national and international journals of repute. And this is my first textbook on microbiology. But before this, I have already co-authored a book on hospital-acquired infections. And also, I've also been involved with all the NACO publications, all their guidelines and the national guidelines they bring about for the anything to do with STD diagnostics. So I have been one of the resource persons and editorial team member. Yeah, that's about me. Over to Dr. Rohit.
Good evening, everybody. Thank you very much, madam. Good evening, everybody. Really thank all the eminent faculty members to have joined in this webinar on Lippincott Illustrated Reviews Microbiology, which is the latest offering from Wolters Kluwer with respect to microbiology in relation to competency-based medical education curriculum that we are soon initiating in our own medical institutes. I am Dr. Rohit Chawla, working as an associate professor in the Department of Microbiology at Maulana Azad Medical College. I've done my MBBS and MD both from MAMC, and subsequently did my DNB microbiology, and I'm a recipient of gold medal for academic excellence in the field of microbiology from NB. I've done my American board in clinical microbiology and a fellow of clinical microbiology from Canada and from UK, diplomat of Royal College of Pathologists. I'm also a certified infection control practitioner, attained my certification again from US for that. And again, thank you once again for sparing your time to attend this webinar on LIR microbiology. Thank you once again.
With this, I request Dr. Sumati to just start the webinar by just showcasing her slides right away. Thank you so much, ma'am.
Yeah. Can I have the sharing rights? Yeah.
Yes, ma'am. You have right now.
Yeah. Please give me a moment. Is it visible?
Yes, ma'am.
Okay. Yeah, good evening, everybody, and welcome all of you. In this evening hour, we are having this session, and I hope to make it worth it for you. I wish to share some information on the diagnostic microbiology during this. Since all of you are either faculty members in microbiology or students of microbiology, this will be an important topic. So, first, we all know that microorganisms are very, very small creatures which cannot even be seen under the, with the naked eye. But they spread over us in many ways because they have been troubling mankind for time immemorial. So, why do they store over us? What is it we have that is difficult for us to control them? It's because of some factors. The thing is, audience, can you please mute yourselves?
Ma'am, please go ahead.
Yeah. So microorganisms are not only invisible, but they are also widely distributed. So when you have an unseen enemy who is also all pervasive, then it becomes difficult to control. And they occur in complex associations, and they have to be grown in artificial conditions. So you have to know all their properties in order to, grow them artificially in the laboratory. And more importantly, they have the ability to develop resistance to drugs. So this is a very important point because we will not be able to control them unless we can address this point. So how are we addressing all of this, all these problems about microorganisms? So the subject of microbiology, I'm happy to say, has really progressed in leaps and bounds from the time, for the past 200 years or more. We all know that in somewhere in the 17th century, Anton von Leeuwenhoek invented the microscope, and ever since there's no looking back. Because after that, the Robert Koch, Louis Pasteur, all of them worked with what limited gadgets were available to them. But in spite of their limited gadgets, they have given us what we have today. So they have started us off on this journey of development as far as diagnostics is concerned. So from the simple microscopy, the branch of microbiology has progressed to culture techniques, and then you have the serological techniques, whether it is ELISA or fluorescent techniques, radioimmunoassays, then the molecular assays, and now we have the MALDITOF using mass spectrometry, proteomics, genomics. Now we are into all of this, and the CRISPR technology. So we have progressed quite a lot in this. So the progress is so much that we have been empowered, and now we can call ourselves the clinical laboratory scientist. And we have made ourselves indispensable to the clinicians, because otherwise, without us, they are just guessing. I mean, this is the microbiologist's point of view. So we must remember that the role of the laboratory tests is not only for diagnosis. It is also for other purposes, some of which I've listed over here. For screening, for example, asymptomatic high-risk groups, example in HIV. And for diagnosis, like in atypical cases, when there are multiple infections or asymptomatic infections, then the clinician cannot do a proper diagnosis. So there the laboratory becomes indispensable to make an accurate diagnosis. And then for monitoring treatment, for surveillance purposes, outbreak investigations, syndromic validation. Here again, the best example for syndromic validation would be in STIs, where NACO has given us the syndromic approach to treatment. But even here, the laboratory is required because periodically you have to do lab tests to find out what antibiotics work and what don't. And of course, the research purposes. And antibiotic resistance is a very big topic again, where the laboratory contributes immensely. So what are the methods available in diagnostic microbiology? Initially, I'll tell you about the conventional methods. I'll just touch upon them and then move on to the newer methods. Conventionally, as we all know, we have direct detection by microscopy, where you look at the microorganism either in an unstained preparation or a stained preparation. And then we have the culture techniques where you grow the microorganism in the laboratory under artificial conditions. And after growing them, you identify them so that a diagnosis can be made and you can give the information to the clinicians. And of course, the antimicrobial sensitivity also has to be done. Or you can do a detection of the antigen of the organism. If you cannot see the whole organism or culture the whole organism, a part of it can be seen detected by antigen detection. Then you have the antibody detection in the blood samples and animal experiments. This is more of, animal inoculation studies are more for research than for diagnostics, but that is another option. And nucleic acid detection where the DNA or RNA is detected. So these are all some of the methods available to us. And here is an example of the unstained direct microscopy. This organism that you see over here, this is the typical jerking motility that is shown by Trichomonas. So this is what you see by an unstained preparation, wet mount. And the hanging drop is another unstained preparation microscopy which we do to look for the typical darting motility to diagnose cholera. And potassium hydroxide mounts can be used for looking at fungal elements. And this is a picture from our own book where you can see the fungal elements. And staining techniques, stained microscopy. This again, we use either the stains can be simple stains like methylene blue, or some of the other simpler staining techniques like the gram stain, Giemsa stain, or the special stains like the fluorescent stain. So these are some of the microscopic methods where we use stained preparations. And gram stain is one of the most important stains. So for the students, I've just shown this slide because here there's a beautiful diagram which shows all the steps of gram stain. This again is taken from our book, and where the steps are very clearly seen. And I always tell my students, a student of microbiology who does not know everything about his or her gram stain and has not done it does not deserve to pass microbiology. It is that important in the field of microbiology. So coming to the culture techniques. Culture and identification, for this, we start from the very beginning, like specimen collection, how to collect the specimens, how to transport them, what processing to be done, and then understanding the growth requirements of the microorganisms. For example, what temperature is ideal, what is the pH required, whether they require oxygen or not, like the aerobic, anaerobic organisms, whether the media they require is simple or they are fastidious and fussy and require some selective media for growing. So this is what culture is all about. It's laborious, it's time consuming, but it is a very specific method, and it is still the gold standard for many of the organisms. And it's not important just to culture the organism and grow it under artificial conditions. You also have to identify it to be useful for the clinicians. So how do you identify it? By looking at the morphology and doing a battery of biochemical tests or other tests by which you can identify them. And then once you have identified the microorganism, again, that is not enough, because the clinician has to know what kind of treatment to give, what antibiotics to use. So for this, we have the antimicrobial susceptibility testing is performed. There are various methods of doing it. Here I have shown again this beautiful diagram here from our book, which shows the disk diffusion technique, where you have the Stokes and the Kirby-Bauer techniques. And you can also do the MIC, or the minimum inhibitory concentration. The strip is shown over here in this photograph, again from our book. Yeah. And then you have antigen antibody detection, which can be done by serological tests, starting from the ELISA, which is well known by everybody, the enzyme-linked immunosorbent assays. And you also have other methods like the radioimmunoassays, the fluorescent methods, and many other techniques. So these were the conventional methods which I mentioned up to now. So apart from this, there is a barrage of newer diagnostic tests that are under our disposal now because of various developments in the scientific world. So now the microbiologist is equipped with so many kinds of varieties of tests that it is quite intimidating and mind-boggling for the clinicians, because they don't know which one to go for and what tests to write. So it's up to us to educate the clinicians on these methods. So the molecular diagnostic tests have now taken over where the nucleic acid is detected, that is the DNA or the RNA. These tests are specific, they detect the specific sequences, that is DNA or RNA, which may be associated with the disease, that is with the microorganism. So when do we use this? This is mostly used when we have to look for organisms which are very difficult or impossible to culture. For example, chlamydia, which is a bacteria that's very difficult to culture, and also Treponema pallidum or human papillomavirus, and also for epidemiological purposes, and sometimes even for the serological assays, when you want to enhance the serological assays by using cloned proteins, or you want to use recombinant antigens, then again you have to resort to the molecular techniques. And another important a very important test is where we have to differentiate between organisms which are very closely related. For example, the herpes simplex virus 1 and 2. So they can only be differentiated by using molecular techniques. So these techniques have become so important now that they are considered the gold standard for some of the organisms which cannot be cultured. Then you have another newer kind of diagnostic technique called the DNA microarray or the DNA chip or the gene chip. It is nothing but microscopically the grouping of the DNA molecules on a solid platform. So these are called as reporters, these spots of DNA. And what you do is to these spots, you add the labeled DNA strands. Labeled means the fluorescent dyes are attached, they are labeled. And these DNA RNA strands from the specimen are incubated with these chips. And then you have a scanner which reads off. So here you can do multiplexing. That means many organisms in one go you can diagnose. So this is another newer method. And then you have the proteomics where the proteins are studied at the cellular level. So now what you can do is even at the cellular level, the content of protein, it varies in the normal condition and in diseased conditions like cancers, genetic diseases, or even infectious diseases. So this point is made use of in diagnosis by proteomics. So there are many, many more newer techniques. I can't go into all of them right now. In fact, the sky is the limit, or maybe the sky is not the limit. So there are many other techniques like the genomics for vaccine designs, genotyping for identifying drug-resistant strains. You have high throughput sequencing, the bioinformational computational genomics. You have biosensors, which can also be used for detecting microorganisms. And then there is this MALDITOF, a fancy word which has recently come into our lives. Here again, mass spectrometry is used for speciating the microorganisms. And you have the latest CRISPR techniques also for fingerprinting in cells. So some of these techniques are in the pipeline, or some of them are well established, but many of them are time consuming and labor intensive. So what do you do? When these tests are time consuming and labor intensive, in today's world and today's generation, we want everything fast, whether it is our fast food, where we want our pizzas and burgers to be delivered within 30 minutes, or whether it is the laboratory tests. So this generation wants everything fast. Fast food, similarly, fast lab results. Sample in, result out. That's what we want. So what is the answer to this? The answer is the point of care tests. Point of care tests is POCT, what we call as point of care tests. What does it mean? It means that these tests are performed at the point of care, not in a laboratory or in a clinician's office. They are performed at the point of care. So where you're caring for the patient, that is at the grassroots level, at the ground level, it can be performed. But these tests have certain conditions to be fulfilled. Way back in 2003, WHO has given the assured criteria. To be called as a point of care test, the test device has to have this fulfill the assured criteria. What is assured? Affordable, sensitive, and specific. That means accurate. It has to be user friendly. User friendly means very laborious procedures are not required, and even untrained or semi-trained people should be able to perform it. And it should be robust and rapid. Robust means it should be stable at room temperature. You don't need stringent storage precautions for it. And rapid means the turnaround time should be very less, within 30 minutes, just like your pizzas. The result has to come in about 30 minutes. And then it has to be equipment free. That means you shouldn't be needing a microscope or an electrically operated centrifuge or some devices or anything. So if all of these are satisfied, then you call the test a point of care test. And that's what the world is all coming to now, because we are all in a hurry to get our results. And the advantage of these point of care tests is that it offers results rapidly, so that diagnosis and treatment can be made in one visit itself. So when the patient comes once to the clinic or the results are also available same day, and the treatment can be provided the same day. They don't have to come back again for results. But one important thing is the quality control has to be incorporated in these tests. And a good example of a point of care test is the lateral flow immunoassay, which is called the LFIA. So that's where it all started. And for detection of either antibodies or antigens. And most useful at a patient's site, this is most useful at the first contact with the healthcare facility. Like, when the patient first goes to a primary healthcare center where they don't have very, big facilities for doing diagnostics, in such places, the point of care tests will be very useful because it provides actionable information. This is a very important term, because actionable means as soon as the results come, we are able to take action in the form of a treatment or, quarantine or whatever it may be. And it reduces presumptive treatment. This is another advantage where the clinicians otherwise would be, it's a kind of a guessing therapy. They put the patient on some treatment and then say, get these tests done. So once the tests are done and the organism is diagnosed, the clinician will change the treatment. But here, if you have good POCTs, the presumptive treatment need not be there. Straight away, we can get the proper treatment given. And it also has the potential to become an over-the-counter device where people can just go and buy from the pharmacies, over-the-counter device. And in the privacy of your home, you can do the test. And so this is the lateral flow immunoassay is a very versatile device for testing. And a few words about the evolution of POCTs and some of the important test devices. That is, the examples I'm giving here is one is the cryptococcus. For cryptococcus meningitis, when you make a diagnosis, especially in HIV patients with low CD4 counts where you suspect cryptococcal infections, you may have to right away do a lumbar puncture. And lumbar puncture is a very invasive procedure. So we were using this latex agglutination test earlier, but now there is an improved version of this where WHO assured criteria is fulfilled. So it has become a point of care test now, where you can just use blood sample or plasma sample to test it. So and this will prioritize for your lumbar puncture whether to do it or not. So this becomes very useful. Here, the ordinary test, which was the LAT, has now been made into a POCT. So also in the HIV testing, earlier, the lateral flow immunoassay, we only used to use whole blood, plasma, and serum. In fact, we still use these. But now oral fluids also can be used for this. So again, here, self-testing is possible, where the patient can do the test in the comfort of their homes. They don't really have to go to a laboratory and get their blood taken and all that. So also in malaria, earlier for malaria, we had no rapid tests, but now the lateral flow immunoassay has been it has evolved and a POCT has been made, although it has low sensitivity and specificity, but still studies have shown that this has been saving lives. So that is more important right now. So we have using. So these are three examples of how the POCT or the point of care tests have evolved. And then earlier, the antigen detection POCTs, they have low sensitivity because the assay limit requires that high concentration of antigen should be there. For example, even the COVID testing, which all of us are familiar now, if you take the antigen test and it may come negative, but when you do an RT-PCR, it comes positive. So why is that? Because the antigen detection, the limit here is that you need high concentration of viruses to be for this antigen test to pick it up. So the answer to this is, instead of antigens, now we are using molecular diagnostic devices for the POCTs also. And in January 2015, the CLIA has cleared the nucleic acid amplification test, the first POCT, and now there are many such in the pipeline. So what is the future of these POCTs? This is a busy slide, but I'll just tell you in brief. Microfluidics, this technology has created a fully integrated device with sample processing, handling, multiplexing. Everything has been miniaturized and integrated. All the steps have been integrated into a single chip or device. That's why it's called as a lab on chip diagnosis. So that is one improvement they have done. And the other future of POCTs is communicability. Now, if you are doing all these tests at home or at the site or wherever it is, what is the importance of it? If you do the tests quickly and you get the results quickly, it's of no use unless that result is put to use, the information is put to use. How is it put to use? You have to communicate the results to the relevant people or the relevant labs or clinics for further action in the form of a treatment or disease containment and whatever it is. So now what they're doing is, the newest thing is they're linking this with the GPS systems, the geo positioning systems. They're linking with GPS, and also some of the results can be converged on your smartphones. So if you can, WhatsApp the result or something to the clinician sitting elsewhere. This has been tried out in Africa, because where in remote places, the point of care tests are done and the results are communicated. It used to take a week or more for the results to reach the lab earlier because of postal or whatever delays. But now, with the use of smartphones and GPS, the results are communicated quickly. So then the action also can be taken quickly accordingly. And the molecular point of care tests, earlier all the molecular tests were, although they were high sensitivity and specificity tests, the cost was very high, the turnaround time was high, the manpower was required, trained manpower, and the equipment used were all expensive. But now, with the isothermal DNA amplification techniques, the picture has changed completely, and you have all of these advantages. It's almost like a near perfect test, where it is highly sensitive, highly specific, the cost has come down, turnaround time is just about 30 minutes or so, and I mean 30, a little more than that maybe, but considerably low, and without use of manpower and expensive equipment. So the POCTs have, and also the added advantage of multiplexing. That means many organisms can be diagnosed, one or more can be diagnosed in one go. So a good example of this is we have for syphilis and HIV, in one test, there is a POCT for that. And so these are some of the future prospects for POCTs, how they can be developed and done. And one last word about the host biomarkers. Now, so far, we've always been concentrating on the microorganism. We want to detect its antigen, its antibody, and then look for the DNA or RNA of the microorganism. But what about the biomarkers in the patient's samples? So when there is an infection, we also know that there are many chemical substances which are produced, and these biomarkers can be detected. And for these also now, point of care tests are being designed, like for detecting CRP, heparin binding protein, the procalcitonin, tumor necrosis factor, and all of these. So for this also now we are getting POCTs. Many of them are not yet FDA approved, but we are getting there. So in the end, I just want to tell you that there may not be one single optimal test for detection of microbial agents. It's not like, a lock with a single key. It's more like a Swiss knife, where you have a whole lot of array of tests which can all be used to advantage in arriving at a diagnosis. So the clinician may ask you, when the microbiologist tells him about all these variety of tests available, he may ask you, how do I select an appropriate test for a particular disease or a clinical condition? So then you have to ask them a counter question. What do you want? You want speed? Then go for real-time applications. You want quantification? You want multiple targets? Use the microarrays or the multiplex PCR. You want viability, or you want for research purposes a low throughput, high throughput, whatever it is, accordingly, you can fine tune and tailor make the test that you would want to use. So this is in a nutshell, what diagnostic microbiology is all about. The future is very exciting because we have a whole lot of things opening up. And with these point of care tests, I hope we'll be making all the clinicians very happy. But lastly, I want to end by saying it is all the cooperative efforts of the clinicians and the laboratory people, because the coordination has to be very good. Otherwise, remember, the laboratory report is only as good as the sample received from the clinician. So I thank you all for your attention. Over to Dr. Rohan.
Thank you, madam, for this lucid presentation on various diagnostic approaches which are available to clinicians nowadays, including the POCTs. Now I'd start with my presentation. Mr. Ravi, if you're there, can you provide me with the sharing rights? Yeah, you've already done. Thank you. There's an important topic or set of topics in immunology that is very challenging, particularly for an undergraduate students. Now that we're all very seasoned teachers and we teach about various aspects in microbiology, one thing which is uh, at least I find very challenging is to teach immunology to students. So herein we've put in a small topic on immunoglobulins and highlighting the important aspects in our book, which would help you enable uh, teaching this topic of immunology, making it a bit easier for the students to understand. I hope I'm audible. Am I audible? Please respond.
Yes, sir. You are
Yeah,
very much, sir.
yeah, thank you. So we'll just make a beginning. We'll talk about humoral immunity, a bit about it. What is the structure and function of immunoglobulins? Certain important terms that we need to teach to our students. That is, what is specificity? What is isospecificity? There's a lot of confusion in these two terms, though they sound very similar, but then they are totally different. And then at the end of it, we'll talk a bit about the effector functions. So I'll just try to keep my presentation as short as possible so that we can discuss some other aspects of the book also. Immunoglobulins, we all know, are synthesized or rather synthesized within the B lymphocytes and then expressed on the surface of B lymphocytes. Each B cell, we are very well aware, synthesize immunoglobulins of a single specificity. We'll discuss about this as to what we mean by specificity. And the immunoglobulins which are displayed on the surface of a B cell are referred to as B cell receptor, in comparison to the immunoglobulins which are circulating in the blood or found in other areas of our body, particularly the mucosa. They're all present in the fluid phase, as opposed to the ones which are immobilized onto the surface of the B cell, where they basically act as receptors which recognize their cognate antigens. B cells, they get activated in response to the antigen challenge, and they get then terminally differentiated into plasma cells. And these plasma cells now not only synthesize the antibodies, but also secrete these antibodies or immunoglobulins within the circulation or onto the mucosa. Antibody is another term which is used very commonly in relation to immunoglobulin. That signifies that the immunoglobulin molecule specifically binds to a particular epitope. So then in that sense, it is referred to as an antibody, but these two terms are very often used interchangeably. We all know antibodies, bulk of them are present in the fluid phase, so therefore they form a very important part of our humoral acquired or adaptive immune response. So this is being taken from a book where it shows the B cell receptor, which is there on top of it, which binds with the cognate antigen. Once it binds with it, it gets activated, it clonally expands and then converts into plasma cell, which then secrete the antibody. Antibodies then covalently bind to their specific antigen and they immobilize that antigen. They may also tag the antigen so that once it is tagged, it is then taken care of by other components of the immune system. So these are the effector functions that we're going to talk about just a few moments from now. As antibodies specifically bind to large number of different types of antigens, and they are virtually limitless antigens which are present uh, in the microbial world, we need to have large repertoire of these antibodies, each binding to those specific antigens. So there's a huge, uh, in high number of specific antibodies which are available, huge repertoire available. Then only we'll be able to bind to the specific antigen, then act against it. Antibodies structurally are basically glycoproteins, so bulk of it is protein and small amount of carbohydrate is also there. But most of the functional aspects of antibodies and immunoglobulins can be explained by the protein structure, and that's what we are going to discuss. Antibodies or immunoglobulins can exist either in a monomeric form or a polymeric form. The monomeric structure is the one that we're going to discuss right now. They consist of two heavy chains and two light chains. The two light chains are kappa and lambda. A single antibody molecule will either have a kappa chain or a lambda chain, but never both together in a single molecule, that is. While heavy chains are of various varieties, these are gamma, alpha, mu, delta, epsilon. And the first letter of this Greek word, gamma, signifies the class of the antibody. So if it is gamma, it's G, alpha, A, mu is M, delta is D, and epsilon is E, so on and so forth. The genes where these these proteins are the genes for these proteins are located are different. For all the heavy chains, the genes are located on chromosome number 14. All the kappa light chains, it is located on 2, and all the lambda light chains, it is located on 22. So for light chains, they are different chromosomes, but all the other heavy chains, the genes are located on chromosome number 14. As I was mentioning earlier, there are 2 heavy chains and 2 light chains in an antibody molecule. So it is a dimer of dimer, and it is a Y-shaped molecule. All the 4 chains, the 2 heavy and the 2 light chains, are arranged parallelly. So for every polypeptide chain, with the knowledge of biochemistry, we know that there's one end which is a amino terminal end, and the other end is a carboxy terminal end. So all the aminos are arranged at the top, while all the carboxys are arranged at the bottom. So all 4 chains are arranged parallelly. Within the both heavy and light chains, we've got a variable region which is towards the amino terminal end, and the constant region which is towards the carboxy terminal end. And that's what defines the specificity. The variable region varies because it has to bind with huge number of different antigens that the human is going to encounter throughout his or her life. So it is the variable region which provides this specificity to the antibody, as you will understand better as we move forward. So why variable regions are called as variable regions? Because the amino acid sequences of the variable regions vary much more than the constant regions. So this was what we discussed: 2 heavy chains, 2 light chains. So within the heavy and the light chain, we've got a variable and the constant region. So we've got 2 VL, variable light, 2 constant light, 2 variable heavy, and 2 constant heavies. As I said earlier, the light chains are either kappa or lambda. A single antibody will have either kappa or lambda, never both together. Antibodies containing both the kappa chains are twice as common as compared to lambda light chains. Another important concept that students often are difficult to understand for them is what is a class of the antibody. So this is defined by the constant heavy chain region. And the gamma chain, alpha, mu, delta, epsilon, all of these are nothing but the different varieties of the CH regions which define the class. A single antibody molecule contains only one variety out of the 5 heavy chains. So out of the 5 heavy chains, only one of the 5 would be present in a single antibody molecule. Then within the constant heavy chain regions, there are various domains which are present. So just to show you one of the pictures of our book, which shows the diagram of a monomeric antibody. These are the heavy chains, 2 identical heavy chains, the light chains. Heavy chains are divided into the variable heavy, the constant heavy, both on both the sides. Light chains, similarly, we've got variable light, constant light. The antigen binding or epitope binding domain is formed by the variable heavy and the variable light, both together associate to bind with a particular antigen. So this is the antigen binding site of an antibody. This is what varies a lot, so that it is able to specifically bind with millions and trillions of different type of epitopes that an individual might come in contact with. Just shows you the domains over here. Just to show that how different antibodies have different specificities. If you can see over here on the left side, we've got a VHBL region, which has got a different conformation than over here. So it will bind to its own cognate antigen and not the other one. Similarly, because of it, the structure of VHBL on the right side will bind to another cognate antigen, not the other one. So this provides the specificity to the antibody. And within the same antibody, there are 2 heavy chains which are similar and the light chains which are similar, but the variable regions of 2 different specific antibodies or different specificities are different. These are the 2 people who discovered the structure of the antibody by breaking the antibody molecule. This is a very common question that we ask our students. Classical experiments performed by performing the cleavage of the antibody by papain and pepsin. So this beautifully signifies what papain does and what pepsin does. Pepsin cuts the antibody above the inter-heavy chain disulfide bond between the 2 heavy chains, giving rise to 2 FABs plus 1 FC. So FAB can be explained as fragment of the antibody which binds with the antigen, has got a valency of 1. So there are 2 molecules, each having a valency of 1. We'll discuss what is a valency. Similarly, we've got pepsin. Pepsin cuts below the inter-heavy chain disulfide bond. This is the inter-heavy chain disulfide bond, cuts below it. So you get FAB bracket 2 with FC dash. FAB bracket 2 means it is a single molecule because it is all held by the inter-heavy chain disulfide bond to give you a single molecule having the capability of binding with two epitopes. So it is single molecule bracket 2 means is able to bind with two epitopes. Here it means 2 FAB means two separate molecules each having a valency of 1. Here it is a single molecule having a valency of 2. So coming to in more detail, what is specificity and isospecificity? A bit of it we've already discussed. So specificity is the antigen with which an antibody binds with. So it is defined by the VH and a VL region. And the number of VH-VL regions indicates the valency. So this is a diagram just showing the specificity as was discussed earlier. Important point that we can impress upon students is that single B cell is monospecific. So all the antibodies produced by a single B cell, all the plasma cells originating from that single B cell will bind to only one cognate antigen or epitope. Isospecificity is another complex concept. These are variations of the antibody, and all variations are present in all members of a species. So as a human, antibodies have got 5 variations, G, A, M, D, E. All these 5 variations are present in all normal members of a species. So in case of humans, all of us, as long as we are normal, we've got all 5 isospecificities, and this is defined by the CH region. Some classes have subclasses like G, we've got 4 subclass, and A, we've got 2 subclass, which calls G1 to G4 and A1 to A2. These classes and subclasses define the effector function. So I'm just going to run you for the because of the paucity of time, the IgM, the effector functions, the IgG, the effector functions, then IgA effector functions, similarly D and E. Okay. So all these are diagrams which are taken from our book. Then we come to effector functions, what these are. So how do antibodies act? Nowadays, there's a lot of talk on neutralizing antibodies with regard to COVID-19. So these are the antibodies which bind to the pathogen and prevent its binding to the receptor. So that's what we want. If you talk about the recent example of COVID, the antibody binding to the pathogen, preventing its attachment to the host cell, so thereby preventing the infection altogether. Opsonization. Some of the antibodies, particularly IgG antibodies, bind to the pathogen, and then the Fc part is recognized by the phagocytic cells by which it is able to phagocytose. Some of the antibodies, particularly M and G, are able to fix complement. They lead to deposition of complement components like C3b, which further enhances opsonization in the same way as IgG, and then formation of membrane attack complex down the line, which creates pore in the cell membrane of the pathogen and thereby kills it. Another mechanism by which antibody is able to get rid of the pathogen is by antibody-dependent cell-mediated cytotoxicity. So antibody binds to the cell surface antigen of a pathogen. The Fc part is recognized by the NK cell. NK cell reaches the pathogen, and the bridge that is formed is by the antibody and releases its substances like granzymes and perforins, which damage the pathogen in question. So these are the various ways by which the antibodies tag the pathogen, and thereby many times with using other components of the immune system, we are able to get rid of the pathogen. Certain rules to summarize: single B lymphocyte is monospecific. So all antibodies produced by single B cell or plasma cells originating from that single B cell will bind with a single epitope. Plasma cell secretes antibody of only one class at a time. However, it can switch class. So initially, it forms IgM, and then it can switch to IgG. So these are some rules with regard to the antibody synthesis. So with this, in a nutshell, we discussed about the structure and the function of the antibody. With this, I'll just conclude my presentation. Now we'll come to just a minute. The next part where we are going to discuss about some salient features of our book.
Yeah.
Can I switch on that presentation, madam? Then we can run through
Yes.
that simultaneously.
Yeah. I'm just. Do you have it or?
Yeah, I have
okay,
it.
I have to have the sharing rights. Yeah. Okay, I'm just switching it on now. Is that visible?
No, it's not yet.
Please unshare it and then share it again, please, ma'am.
Or shall I share it, madam? Mine is a.
How about now?
Yeah.
Yes,
Yeah.
ma'am. Very much. Thank you, ma'am. Thank you.
Yeah.
Yeah. So initially, if I may just say so, when we were approached to write this book, the Lippincott book, I'm speaking for both Dr. Rohit and myself. We were extremely. It was quite challenging because Lippincott is a very well-known name, and for us to be, writing the South Asian edition, which is, I think, the first time that it's happened. It's the first edition of the South Asian version. So when we were approached to do the writing of this, it was a bit challenging, but then highly exciting also because, after all, getting yourself associated with a name like Lippincott Illustrated Reviews, which is a very well-known name. So we sat down to it and then we worked very hard on this. And the result of it is what you have here in front of you. I think many of you may have seen the copies also. So we would like your feedback on all of this. So, sorry, just one sec.
We got on to the next page. Yeah.
So this is the South Asian edition has been written by Dr. Rohit and myself. And this was launched last year in the month of November. We even launched it in one of the medical colleges in Delhi, Maulana Azad. And the details of the book, I think, as and when you go through it, you can just see also. We, both of us, we've already spoken about ourselves. We have a rich experience of teaching because both of us belong to very well-established medical colleges where we are working as faculty. So we're teaching not only the MBBS students, but also the postgraduate students where we have our own thesis candidates and all of that. Apart from this, the dental, BSc, MSc, and all that. So these are the author details of Dr. Rohit and me. Yeah, Dr. Rohit, could you?
Yeah, thank you. So these are some of the unique features of our book which I'd like to highlight. So it's a book which emphasizes on the clinical aspects of microbiology. This is one thing which is lacking in many of the older books. So we try to give a clinical context in as many situations as possible. So it is in fact full of clinical vignettes, how you apply the knowledge of microbiology in a clinical situation. Perfectly, this then aligns it to the competency-based medical curriculum where you want the Indian medical graduate to link a particular subject, particularly in relation to pre and paraclinicals, to the clinical outcome. Complete coverage of various subdisciplines of microbiology, namely general microbiology, immunology, bacteriology, virology, and so on and so forth. Hundreds of MCQs and short and long answer questions have been provided at the end of the book. New chapters have been added to make it relevant to the South Asian countries, particularly the largest one in this part of the world, that is India. Updated scientific names of microorganisms. We all are microbiology. We know every year new names of the microorganisms are added to make life difficult for not only us, but more so for our students. So just to give an updated list of these scientific names. Color-coded schemes, which I'm going to discuss later, how it has been incorporated in this title. Disease processes has been explained. That's, I think, a fundamental feature of this book, where a lot of pictures have been used to explain the pathogenesis. So we'll again discuss about it later in the presentation. Large font has been used so that it's easy for the eyes of the student. Pathogen summaries have been provided. Extensive cross-linking has been done. Quick review of clinically important microorganisms, disease summaries, and illustrated case studies are a few of the unique features of this book, which we are going to detail in subsequent slides. Next slide. So as I was talking about the clinical emphasis, that's what we really want our students to actually know. Gone are the days that you just talked about the laboratory tests and the microorganisms. How it relates to the clinical situation is something that is the leap that we need to take now more so ever. Next slide, madam.
Yeah.
This is a new section of immunology. If you've gone through the older version of LIR microbiology here, it really is a major improvement where the whole section of immunology with beautiful pictures, which I just showed, one of the smaller topics that I covered in on immunoglobulins. It is covering a large number of aspects of immunology. Immunology is one subject where you cannot teach unless you are able to show some good diagrams and figures, which actually make the comprehension easy for the student, for an undergraduate student particularly. We've been reading microbiology and immunology since ages, and it becomes often sometimes difficult for us also. So think about the plight of a student. So it is ideally, I think, suited for a undergraduate student to be able to understand. So chapter 6 to 13, large number of immunology topics have been covered. So it's a fairly comprehensive coverage that we've provided with regard to immunology. Next slide, madam. Just to show you some beautiful pictures that are incorporated to make the comprehension easier. Next slide, madam. So one of the other pictures, scores of pictures are available in the title. If you go through it, you'll realize it's not only one or two images, scores of images which are available. Next slide, ma'am.
Yeah, so we've, as Dr. Rohit was mentioning, we've added a whole lot of new chapters, considering what is important from the MBBS curriculum point of view, as well as the kind of questions that are set in the Indian setup. So the previous edition of the LIR microbiology, that is the other foreign version, it did not have certain chapters which were relevant for us. As Dr. Rohit pointed out, the immunology section was totally new because they had a separate book for immunology. But here we've made it all a single go-to book, where we've added the immunology chapters here. Also, some of the important chapters like sterilization and disinfection is every microbiology student during the exams will always get some question or the other from this chapter. It's one of the most important chapters, sterilization, disinfection. So we've added that in our in this edition. And also the air, water, milk, and food microbiology. This chapter has again been added as a new addition in our book. And healthcare waste management. Now, you know that all of us are well aware that during our MBBS days, biomedical waste was not such a big thing. In fact, there was not even a single class on that. But now this is being taught in microbiology. There are whole chapters dedicated to this topic. So we have also added a whole chapter on biomedical waste, which in the form of healthcare waste management. And there is a new chapter on bioterrorism also, which was not there earlier, which we have added. And the chapters have been extensively revised. Apart from adding these new chapters, even the older chapters which were already existing in the other LIR microbiology, we have revised them extensively to make them relevant to South Asian context. Because, for example, if you take the newer diagnostic methods for infectious diseases or mycobacterial infections, for example, tuberculosis is a huge problem in India and Southeast Asia, say Bangladesh, India, Nepal, and all of these countries. Whereas in the West, it may not be that important. So when we saw that the chapter on tuberculosis was inadequate, I wouldn't say inadequate, but it was not that relevant probably in the other edition. But in our edition, it becomes very relevant, tuberculosis, leprosy, all these chapters, because these diseases are still prevalent in our countries. So we have picked out these topics and made them more elaborate and in a lucid way manner. We've tried to write them so that the students will, what is relevant to our countries, that will also be studied. So also in hepatitis, HIV, AIDS, sexually transmitted infections, all of this. Yeah, Dr. Rohit.
Yeah, I was just referring to the color-coded schemes for an undergraduate student. I think microbiology is one subject which seems a bit monotonous. So just to add color to what is presented, I think will give it a visual appeal that an undergraduate is always looking for. So just to give you some sense of what we've incorporated. So the classification of, can you just go back?
Sorry. Yeah.
Yeah. So just to give an example of classification of bacteria, we've divided into the ones which are lacking cell wall, having a rigid cell wall, flexible cell wall. So you can see these color codings. So it is surrounded by yellow, it is surrounded by green. So these are the color coded schemes which are used all across when you actually cover the chapter. So it gives you a kind of a visual impression. Further subclassification of rigid cell wall microorganisms, bacteria into the ones which are unicellular, filamentous, the ones which are unicellular, obligate, intercellular. All throughout the book, all those microorganisms which are obligate intracellular would be having this pinkish uh, border around it. And then we are further subclassifying the free living ones into gram positive and gram negative. And obviously, you would agree gram positive would obviously have to be kept as blue and the gram negative as red. So, so on and so forth. Just to give you a brief idea as to how it makes a visual impression when you are reading the topic and how you can relate it all down the line when you're reading that topic in further detail in subsequent chapters. Next slide, madam. Yeah, the same thing has been done for viruses. So you have DNA, RNA viruses. You've got single-stranded, double-stranded, DNA further again, single-stranded, double-stranded. So the ones which are enveloped in each of these categories have been given a green border. So these are little things which we've tried to incorporate. Next slide, ma'am. Here you get an extensive cross-referencing of all the major bacteria. So right at the beginning, this is one of the initial chapters. In fact, it is chapter 1 only. So just to give you a visual impression to the student as to how they can actually navigate through various chapters in bacteriology. It becomes very easy for them to navigate once they go through the whole text and then they want to read a particular bacterium. By seeing their properties, they can immediately go to that chapter and read it. Next slide, madam. The same thing has also been done for viruses. Again, the color schema which is being used is presented over here, and then each of these chapters have been cross-referenced in each of these categories, and immediately a student, if would want to go to that particular chapter, would straight away go over there without any fuss. Next slide, madam. These were the updated list of scientific names of the microorganisms that I was just referring to. So all these older names, the newer current names have been given, so that a student is not is familiar with the new names, but also is familiar with the older names, because many of the older textbooks still follow these older names. So therefore, to avoid that confusion, I remember my year as an undergraduate student, it was sometimes very confusing to know that bacterium is known by two different names, and eventually when you study it and realize that it's actually the same thing only. So it is better to give it right in the front so that they realize that, okay, we are talking about the same bacterium. It has undergone a taxonomical change and a name in its nomenclature. Next slide, ma'am.
Yeah, Dr. Rohit, just to uh, say something in this context. For example, the Pneumocystis carinii, we still know it as PCP. Pneumocystis carinii pneumonia. The word has stuck into everybody's minds. Even the clinicians, they say PCP, that is Pneumocystis carinii pneumonia. But now the word Pneumocystis carinii, the name has changed to Pneumocystis jirovecii. So it becomes a little confusing. So we need to know the old name as well as the new name. That's what I meant. Yeah.
Yeah. Again, for an undergraduate to make this a bit of a drab subject interesting, there's an embellishment of cartoons which have been given at appropriate junctures. Just to give you one of the images of these cartoons in immunology, how the diversity, I was just talking about immunoglobulins and the highly diverse is created. So it is just given in a cartoon kind of form so that it really drills into them as to how diversity is created by giving an everyday example. Next slide, ma'am. Another of those cartoons, you got those Sherlock Holmes and how the immunosurveillance works. So just to give them a bit of fun kind of a thing and attach it to a more serious concept would definitely, we believe, increase the retention power of the student. Next slide, ma'am. Another of the unique feature of this book is the classification that has been given. So we are not into a very formal taxonomical classification, which is really a put off for a student. We want to give them a classification which is easy to understand and it is relevant in their clinical workup later on in life, rather than talking about details of taxonomic classification, which is a bit tedious and nobody remembers at the end of the day. If you ask me, I also don't remember the taxonomic classification. So free living bacteria, if you see a gram positive, it's a coccus next level. And what are the important species within it? Similarly, for DNA virus, double-stranded envelope, these are the viruses which are given. And you can see there's a small S which has been written alongside it with circle. So this actually gives you the summary of that organism later on in chapter 47. So all those important pathogens which we believe are important for an undergraduate to know have been put as an you can say an asterisk with an S, and all these with an S means they have been covered as a summary later on. We'll talk about or dwell on that just a few moments from now. Next slide, ma'am. Another of the unique features of this book is a huge number of photographs and collections on pathogenesis. So here, just to show how a latent tuberculosis turns into an active tuberculosis, a tubercle is formed and later on it gets reactivated and forms a cavitary lesion. So just to show the progression of the disease as it happens, and you'll find it being given in many of the major pathogens that have been discussed in the book in a pictorial form, which makes it easier for a student again. So the whole idea of this book is to make it student friendly, and also for a teacher to be able to teach it in a very easy and a friendly manner to the student, so that it is at the end of the day understandable to an undergraduate. Undergraduates, we all know, are burdened with so many other subjects, so they want something which is easy to understand, easy to retain in the longer run. Next slide, ma'am. I was talking about the summary of bacterial pathogens. Again, this is a unique feature of our book. Next slide, ma'am.
Yeah.
So, next slide, madam. So major pathogens, at the end of the chapter, we've given the clinical manifestations pictorially associated with the laboratory methods used for their diagnosis, again pictorially, along with the main antimicrobial treatment of choice for these pathogens for a particular clinical condition. So this is given for staff over here, and subsequent slide would, I think, give the similar details for Neisseria. So similarly, you get these clinical picture, you get the microbiological diagnosis, what are the methods available, then what is the treatment which is available. So this again has been given at the end of every major chapter, not in all, at end of major chapter, just to summarize and give a clinical context. Again, adding on to the retention of the student and the clinical relevance.
Yeah, another point here is just before the exams, sometimes the student would have read the entire chapter earlier, but at the time of exams, on the last, the day before the exam or something, say, so they cannot, they don't have the time to go through the entire chapter. Then these summaries are very important, because once you read these and these pictures in your mind, they help you retain and reproduce it in your exams, So that's another added point. So,
Again, we use a large font size for the student to be able to capture the information easily. So I personally felt that the too small a font size, it becomes very difficult for a student to be actually scanning through the text and then taking out the relevant information. Adequate space has been provided on the sides on each page to jot down notes. As we were talking about the connect with the Indian context, all the important national health programs which are in relation to infectious diseases have been covered and duly referenced, like National AIDS Control Program, National Vector-Borne Disease Program, so on and so forth. Next slide.
In fact, this one point, the space provided in the slides, this was pointed out to me by one of the faculty members who went through our book, She called up and told me that she was very happy with the space that's been given on either sides of the book, because you can jot down some notes or some last minute, which you would like to read later. So that's a very, she said this has been very thoughtfully done, So then it struck me that.
So this was in microbiology. If you talk about the clinical context, we have to go back and forth because there are large number of organisms we know which cause the same syndrome. So extensive cross referencing, just take an example of Microbiology genitalium, which is again one of the important causes of PID. So how to link it with the other major causes like Neisseria gonorrhoeae and Chlamydia trachomatis, sorry. So we've cross-linked with those chapters so that it is easy for the student to navigate back and forth if a particular clinical syndrome is being covered, which can be caused by different other pathogens. So similarly, it has been done for other pathogens, which all cause a similar kind of a clinical symptomatology for the student to be able to actually go back and forth by giving these extensive cross-referencing within the text itself. Next slide, ma'am, if you don't want to add a point. Yeah, so again, how the national programs have been referenced wherever appropriate, just to give example of TB control program. Next, madam. Just to add on to the visual appeal, we'll just cover some of the important points, ma'am. Can you just go on to the next slide? Visual appeal, we've been talking and we've had many examples. So let's talk about some of the important multiple choice questions. A lot of clinically oriented questions have been added. I mean, gone are the days that in our times we were asked a straight factual question. Nowadays, we get a clinical history. We have to assimilate information in the clinical context, arrive at the appropriate methodology that you are going to use for diagnosis as far as microbiology is concerned. So in fact, more than 80% of the questions that you're going to find at the end of the chapter are clinically oriented questions to give that flavor. And nowadays, in the entrance exams for PG, we get clinical questions. We don't get those, hardly get those questions which are straight factual questions. So it really allows a student to synthesize the knowledge and apply that knowledge as he or she would do in a clinical setting. So we want that to happen for a particular student and not really mired with only the technical knowledge of microbiology, but how to apply it. Next slide, madam.
Yeah, and also the answers are given here. It's not only the questions, so they have been answered also.
Yeah.
With
So
the
next slide, I'm going to show
explanations.
Yeah, we're going to explain. So just to give you, as madam was mentioning, so the question is there, the choices are there. We've given a detailed answer to that question. So how that particular the correct choice is correct and why the distractors are incorrect. So distractors you can see as your differential diagnosis in a clinical setting. So these are what the distractors you're going to get clinically, and this is what we have tried to add in the question to make it more clinically relevant, because it's what the distractors which actually teach you than the actual answer. So we've given a comprehensive answer to that question, telling why correct is correct and why the distractors are incorrect, which further adds on to the knowledge. A lot of short answer, long answer question. The MCQs basically help you for your entrance exam. But ultimately, you have to answer your questions in your university exams. So university, most of the exams which are being conducted contain short and long answer questions. So therefore, we've tried to cover that also. But nowadays in CBME, there's some percentage of questions which will be MCQ based. So covering all of this holistically, short answer, long answer, MCQs, not only the student is prepared for the university exam, but also later on for their entrance exams. Next, madam. Just to give you some glimpses of the short and long answer questions. Next, madam, please. Hundreds of clinical images. Nowadays, what we get is that NEET exams for PG entrance, image-based questions are asked. So with the text, you get image or only image. And then a little bit of clinical history is given. So the scores of images, hundreds of images that have been included definitely would help the student to cater to that particular requirement. Next, madam.
Yeah, in fact, I must share here that Dr. Rohit and I are personal, are the photographs which are actually clinical examples. These photographs have also been added in them. So from our personal clinical images.
20 years or so of clinical images that we've collected all those years, we've tried to incorporate it and try to make it relevant, particularly from the Indian standpoint. Next, madam. Just to give you a glimpse of the images that have been added and how it correlates with the microbiological findings. Now, this set of chapters, we found it to be very interesting, or we think that would be very interesting for a student, especially those students who do a last minute reading and go for their exams. So they are kind of, you can say, cheat notes for the student. But anyway, so this gives you a quick review of clinically important microorganisms, particularly certain important bacteria and viruses. So if we can, ma'am, go to the next slide, we can show to the audience what do we mean by it. So this is there given in chapter 47. So some important organisms, the name has been listed, what are the various species which are there, pathogenesis and clinical significance, treatment, prevention, and laboratory identification. So quick, ready reckoner for the student at the end of studying microbiology, going for an exam, jittery, cannot read the whole subject, quickly can run through these kind of notes, which are just a summary of what has been extensively covered in the dedicated chapters, just for the quick review and just to assimilate finally before going in for the exam. Disease summaries is another new thing, which again is in consonance with the requirements of competency-based medical curriculum, where we assimilate all the major microorganisms causing a particular disease at one place and give it visually to a student. So let's say if a person has to answer question on sexually transmitted infections, UTIs, meningitis, so different chapters have been given. In each of these topics where the pathogens have been covered and their major features have been covered in a clinical context in a syndrome-based approach. We'll just give you an example as we move forward. Next, ma'am, if you can go.
Actually, here I recall my own student days where the textbooks had only, if I had to go make a list of all the urinary tract infection causing organisms, I would go to individual chapters. I used to underline them and then make my own notes. So here it's all, it's sort of ready-made.
Yeah. So just to give you a glimpse for bacterial meningitis, the major bacterial causes have been listed out. Again, we use the same color-coded scheme for the student to understand which one is the most important as far as our context is concerned. What are the clinical symptoms, again, in the form of small cartoons so that they can retain.
Yeah.
Next.
Here, if you can see, if all of you can just take a look at A, I just want to direct your attention there. It says overview of common causes of bacterial meningitis in adults. So there you can see the large blue arrow. That means Streptococcus pneumoniae is the most common. So just by visually looking at it, you will be able to enumerate it in the exam. You can write that the commonest is Strep pneumoniae, and then Neisseria meningitis and Streptococcus. So depending as per the importance, they have been given over here in a very nice and diagrammatic way.
Even if the child is not aware of the percentages, we really don't want them to to cram percentages. What we are more important is for them to know that a particular pathogen is the most common cause. So I think that's much more important than to actually rote learning and which percentage is higher and which is lower. I think doesn't make sense at the end of the day.
Yeah.
That's another unique feature of the book where we've given the real life scenarios in the form of case histories, and then these case histories have been followed up with series of questions one by one. So gradually, as you keep on answering those questions, a bit more information is unraveled, a bit more with the second question, a bit more with the third question, and ultimately you arrive at a diagnosis. And as what you do clinically when you practice microbiology has been given in the form of illustrated case histories. And I'm sure this will be very useful not only for the students but for the teachers to bring home the point how microbiology saves lives, how at the end of the day it is the difference between life and a certain death. So just to give you one of the examples, it is there in front of us. A question has been given, a case has been given, and then you will find series of questions, and gradually the information builds up and we arrive at a diagnosis. And all of those techniques the student has learned in the rest of the book is being used over here to arrive at a logical conclusion as to the etiological diagnosis. So I think that's about it. Lastly, can we show the competency-based, ma'am, how the mapping has been done? Because that would be the most important thing for our esteemed teachers to take home.
Yeah, I'll just take a minute for this. So meanwhile, if you can just...
Yeah. So you must have seen the competency-based medical curriculum, which has been launched by MCI, where a large number of different competencies have been given. And these competencies not only relate to microbiology, but as an integrative concept, they've been uh, amalgamated with other subjects like in pathology, community medicine, internal medicine, pediatrics, so on and so forth. So what we've done is that we've laid out all those competencies and how they relate with the individual chapters in our book, and how you'll be able to find that information at the end of the day when you are going to develop those competencies, when you actually get down to teaching those competencies to the students. So I'll just request madam to please bring on that slide, or else if it is difficult, I can bring it on from my end.
Can you just tell me what it was called? RC. Okay, I got it. Yeah.
It's not getting projected, madam.
Yeah, I'll just do that.
Yeah. Yeah. I think, yeah. So if you can increase the size. So this, I think, I believe would be very useful to all our teachers out there. It's a very extensive document that we've really spent a lot of time wherein what we've done is that each of these competencies have been listed and how it relates to the individual chapters, because now what we're going to teach is the clinical microbiology, wherein one particular syndrome, as I was mentioning earlier, may be caused by a large number of different microorganisms. So therefore, you'll find it very easy to then relate and collate the information at the end of the day in relation to each of these competencies. Just we have listed all these competencies, described those competencies as given by MCI, and then given the chapters, and not only the chapters, but also the individual page numbers within those chapters where you'll find the relevant information to collate that particular information before you go in for your session with the undergraduate students.
Yeah, and this time, in microbiology, it's general microbiology and immunity is covered as one topic, and after that, the system-wise, CVS and blood. Then you have the gastrointestinal hepatobiliary system. Then you have respiratory system, musculoskeletal. So as and when they have given the, what the student is supposed to know in these topics, so that we have mapped it out and put the chapters and pages accordingly. So this will be very useful even for the clinical, postings of the student, because these are the kind of conditions, clinical conditions they are going to see in the wards and everywhere when they go for the clinics, and they can get back and correlate with that with the textbook.
So we really don't foresee this book as a book which is for one-time use during that prof only. We foresee this book to be used in their clinical postings also, where they can go back and see what they've learned earlier and try to correlate with it. And that's what actually at the end of the day, competency-based medical education is all about, to integrate various facets of medicine holistically so that it can be used in the diagnosis and treatment of an individual patient.
Yeah. And there is also a section here where it is integrated with other subjects, with other subjects like biochemistry, pathology. So if you go into the, curriculum, see the correlation with other subjects also, pharmacology. So these also have been addressed, community medicine, dermatovenerology. So that's how we've done it, according to the same questions that are given in the curriculum-based learning. So we have incorporated the same thing, and each of them has been painstakingly addressed with the chapters and pages. That's it, Dr. Rohit.
Yeah. Thank you.
Well, thank you, Dr. Sumit and Dr. Rohit. It's such a fabulous webinar, I must say, where you have added a lot of content. Thank you so much for that. Also, just to begin, because we already extended this time, but I have a few questions right now, so we will just answer those questions. The question with Dr. Avinash, he's assistant professor of microbiology. He's saying, can you please highlight the clinical implications of IgE and IgG with a clinical case-based question?
Sure. I think the point is very well taken. maybe if you can go through the book, there are clinical scenarios which have been given, not particularly related to immunoglobulins as a chapter, but if you further go down the line, you'll find many clinical scenarios in immunology which have been given, particularly if you talk about IgE as one of the audience has mentioned, how it relates to the type 1 hypersensitivity reaction. The whole section has been given and MCQs have been put in that particular section. Similarly, IgG and how deficiency of IgG leads to an immunodeficient condition has been provided in a separate chapter on immunodeficiencies. So, as I said, immunology is one topic which is totally interrelated. Though we teach them in different levels, but ultimately you have to synthesize it at one place because everything is connected with the with the subsequent texts in immunology. So if you would read the whole section on immunology, I'm sure you'll definitely be able to find what you're looking for and make use of that in teaching your undergrad students. But your suggestions are very welcome. And definitely, if something is amiss because of our oversight, we'd take your suggestions, try to improve upon them in our subsequent edition.
Sir, we have another question.
It's our honest request that people who go through the book, please give us your feedback so that we could improve things in the next subsequent episodes.
Perfection is one thing that does not have a finish line, so we try to attain it but never achieve it. But then you will help us as esteemed faculty of one of the important medical colleges in our country to give us a feedback so that we can further improve upon the content and make it even more relevant in a subsequent edition. We really love that.
Thanks, Dr. Sumit, Dr. Right. One more question, sir, is there. So they are asking, can we please discuss clinical implication of molecular assays with the help of a case study for any one infectious disease?
I didn't get the question, please.
I'll say clinical implicate. Can we discuss clinical implications of molecular assays with the help of a case study for any one infectious disease?
See, molecular assays, actually, there are a whole lot of them. So molecular assays, you have any clinical condition, matlab, you want for some particular disease, is it? Yeah, see, even if you take HIV, in HIV, they're doing the viral loads. These are all molecular study based only, they are all
yeah, ma'am. What we've done is that we've added the relevant molecular assays with regard to a particular pathogen and tried to explain what is the relevance of a molecular assay particular to that pathogen, because different molecular assays are used for different purposes for a different pathogen. So wherever relevant, we've tried to give that information, but if you want it to be given at one place, definitely we can look into and maybe
yeah, the comments
in
the topic in chapter 4, we've given the diagnostic microbiology. So there we've covered the molecular. I've only taken a part of what we've covered in that chapter. Only a few things I've quickly gone through them, not in detail. But if you go to our book, we have written about the molecular diagnostics, how and all the the recent molecular diagnostics that are there with examples. So you know in which clinical conditions these tests assays are being used.
Okay, ma'am. There's another question which is, please discuss about immunoglobulin genomic diversity.
This is a very important question, but what we perceive is that this question is more to do with a postgraduate level. So we have not dwell on it too much because it will be not relevant as we see if we go through a competency-based medical education curriculum for an undergraduate level. But definitely it is a very important topic for a postgraduate. We've given a few hints of how the diversity is created, and one of the cartoons that I'd shown basically gives an idea for thought for an undergraduate level. But purposefully, we thought that it will be too much of an information how a genetic recombination occurs to generate diversity for an undergraduate level. So we've not given too much of an information at this point, but maybe if the suggestion comes in from all of you as eminent faculty, will definitely further give you more details in a more lucid and a simple language for an undergraduate to understand.
In fact, just one point over here. I think in some of the topics chapters, we have made references to the other LIR books, the other books like immunology. For further reading, the student may read this biochemistry and all that, the LIR biochemistry. So we've made references to that. So only put what is relevant from the MBBS point of view. So if a student wants to read more about it, they can go into the other books. We have given certain references in some parts.
Yeah. And we've extensively reviewed the curriculum, which is CBME. And we've seen that some topics which are too heavy for an undergraduate, we've kept them in a small passing reference rather than giving too much of a detail, because it then shifts the focus away from the main topic, which we really want to focus on, that an undergraduate should know these many things at least. There is no limit to knowledge, but then, as Madam said, we've given cross-referencing and further books that can be followed. But if there is a demand, definitely we're going to add it on.
Thank you, ma'am. Thank you, sir. So another question which is there is clinicians might not be knowing these new names, that too if they give culture reports by Vitec. How to overcome these problems? Under what circumstances we can consider some of the rare isolates as definite pathogens in cultures? It's like bacterial or fungal.
Ma'am, would you like to take this question?
Yeah, actually, what the Vitec gives out, the species, if they are very vague or something, it's the microbiologist's prerogative to tell them when they give a report, there is always a footnote or something attached to get the relevance of it also. And the clinician will always have to discuss it with the microbiologist, because the clinicians may not know about the relevance of these things. So it's always by mutual discussion, such things. If there is something very rare, I'm talking about, you asked about rare diagnosis, isn't it? So
Another question is that.
Just
So
one point I'd like to add. I think this is an important starting point. So only when we make aware our undergraduates about the new names in due course, these things would be percolated down when they become big clinicians.
Sir, there's another question is, does it have AET ATCOM module also? AET COM?
ATCOM module actually is for attitude and medical ethics. That is a separate module which is beautifully brought forth by MCI. And that is a self-sufficient document where the histories have been given, particularly in relation to attitude and ethics. So I think there is, as they say, there's no point reinventing the wheel. So if you can go through that ATCOM module, it is a very well-written module which can be practiced. And really, if you were to add the ATCOM module within this text, this would have taken away from the main core area, that is how to teach microbiology to undergraduates. It is very well given in as a separate module by MCI. So I would request you to please go through that, and that is an amazing document. And I think it is so well laid out that by incorporating it, we would not be doing justice to that.
Okay, ma'am, the last question was not actually to, I think, be to us, that okay, is it copy is available online? So yes, the copy is available online, also to the print as well as digital media. You can also buy the e-version of the book, the print version of the book, as well as the books online, also at the campus bookshops. So I think this, I believe, the end of the thing, because I believe my participant also want to know more about it. But I would like to thank you right now, Dr. Samuri, Dr. Chawla, that they have come all the way, in this busy time. Bachelors are the busiest people on earth right now during this pandemic that we have.
It's more so for our eminent faculty who've found time from their busy schedules to be
It's
really heartening for us.
I was coming to that only.
Yeah, and I must also thank the participants. I see many of them online now, especially on a Monday, the beginning of the week, and after your hard days schedule in the hospitals or wherever you're working in medical colleges. So coming home, and then, probably you just had time for a cup of coffee, and then you had to sit for this. So I thank you all for sparing your time to listen to us.
Yeah, and it just shows your commitment as teachers.
Sure, sir. I was coming to my faculty's part only that yes, they are very thankful for coming to and join this webinar. I would also love to thank our commissioning person from the Walters Kluwer, Dr. Vandana Mittal, who's also as a panelist today. who has,
Yes.
I believe, really worked together well in both of you, and brought out such a fantastic book for the students and faculty. So again, thanks everybody for coming onto the board and be a part of this webinar. We promise that in the future we're going to have more webinars, coming along. Maybe we have another webinar on microbiology itself by next month for faculties as well as students. Let's see that. So again, thank you so much. Have a good day, and let's hope to see you all together again on the same platform.
Yeah.
Thank you. Thank
Thank
you once
you
again
so much.
for attending this webinar. Thank you.
Thank you, Dr. Vandana and Dr. Rohit.
Thank you. Yeah,
Thanks.
thank you, ma'am. Thank you very much, and thank you to the whole team of Wolters Kluwer to enable this webinar.
Yeah.
Thank you so much, ma'am. Thank you so much, sir.
Thank you.